My name is Pulendran. I am a professor at the Stanford University School of Medicine. I'd like to talk to you today about vaccines. Now, of course, vaccines are known to be one of the most cost-effective public health tools in history. On that slide, you can see the number of lives vaccines have saved against infectious diseases. It's really been a remarkable success story, a public health success story. And of course, today, given the COVID-19 pandemic, we're all interested in vaccines. Terminologies such as antibody and T cells and neutralizing antibodies, which, at some point were confined to the scholarly textbooks of immunology, have now become commonplace use in everyday vocabulary. So everyone is interested in vaccines. Now, if you look at the history of vaccines, the impact that they've had can be illustrated in this slide, where we're looking at the average life expectancy of humans since the dawn of humankind 3 million years ago. And you can see on the slide that the average life expectancy has hovered at around 25 years. Now, remarkably, around 1796, something dramatic happened. And you can see there what happened in 1796 was Edward Jenner did this amazing experiment in human immunology. What Jenner did was to inoculate this young boy, James Phipps, with pus taken from the pustules of milkmaids who had contracted cowpox. Now, cowpox is caused by a virus very similar to smallpox, except that cowpox causes a much milder version of the disease than smallpox does. So what Jenner did was to inoculate this young boy with a little bit of pus taken from the pustules of milkmaids who had cowpox, and then some weeks later, he deliberately infected this child with smallpox, and he showed that this child was protected from smallpox. So, of course, this experiment would never be approved today by our regulatory authorities, but nevertheless, this was an experiment that Jenner did back in 1796, and in a sense, this was the dawn of vaccinology. So this single experiment in human immunology really resulted in this field that we call vaccinology. Now, the impact vaccines have had on life expectancy is shown here. Shortly after this experiment, you can see there was this incredible increase in the average life expectancy that's continued to today, and today we have on average about 85 years or so. Now, I'm not saying that vaccines have been the only reason for this increase in life expectancy, but certainly vaccines have contributed in a major way. And of course, Jenner's legacy is shown here. In 1980, the World Health Organization announced that smallpox had been eradicated from the planet. So it was the first disease to be completely wiped out from the planet. Such is the power of vaccines. Now let's talk about COVID-19 vaccines. Here we are some 15 months into the pandemic, and fortunately there's been incredible progress in making a whole array of different COVID-19 vaccines in such a short time. And, there are some 180 plus candidates that are being tested in animal models, of which the New York Times COVID vaccine tracker reported that there are now 7 vaccines approved for use in humans. That's all happened in a period of less than a year. That's just been stunning progress, because typically to take a vaccine from a concept to administration to humans, to approval to be given to humans, typically takes years, 10 years, and sometimes as long as 15 years. So what's happened over the last year has just been a remarkable triumph and an incredible success story. So let's talk about these vaccines. So what are some of these COVID vaccines that people are getting? I'm sure you've all heard of the messenger RNA vaccines, shown there on the bottom right. Now, this is a very new kind of vaccine. It's never been used in humans before. So what it is that it's a single messenger RNA, meaning it's a nucleic acid stretch that carries it's a messenger. It carries the genetic sequence for the coronavirus spike protein. Now, this coronavirus spike protein is what you call the antigen. This is the bit of the COVID-19 virus that you have to inject into a human in order to elicit effective immunity and antibody response in that person. So what these companies, Pfizer and BioNTech and Moderna, have done is to construct messenger RNA vaccines that encode the COVID-19 spike protein that can now be injected into humans. And I was very lucky to have received one of these vaccines just recently, and I hope that you all have the opportunity to get either one of these messenger RNA vaccines or any number of other excellent vaccines that are being developed. I'd also like to highlight the recombinant vector vaccines, which are shown on the bottom left. Now, essentially, this is a virus or what you call a viral vector in which you have inserted a genetic sequence for the SARS spike protein. And then you inject this virus into a human. This virus can infect cells of the immune system, and the spike protein gene can now be transcribed and translated to produce the spike protein itself, which then evokes an immune response. And in a sense, these viral vectors are like Trojan horses, if you will. The vector is a horse, it carries the message. And so the immune system is tricked into thinking that it has been infected by the SARS-CoV-2 virus, when in point of fact it hasn't. It simply has been infected by this recombinant vector that happens to have this SARS spike protein. Now, there are many other types of vaccines, and I'm not going to dwell on them here, but just to give you a sense that all these different types of vaccines, including things like the live attenuated one shown there on the top left, which is really a live virus, but considerably weakened so that it doesn't really cause disease, but it is so immunogenic, meaning it can evoke such a strong immune response, and that is the one that Edward Jenner had made, and there are some other classic examples. So you can see that since Jenner's classic experiment 200 years ago in human immunology, the field of vaccinology has been incredibly rich and vibrant. There have been many, many different platforms that have been developed, culminating most recently in the mRNA platform. Now, let's talk about how these vaccines work. Now, I mentioned at the beginning, we've all heard of antibodies now. Even my mother and my grandma, they asked me about what neutralizing antibodies are. The fact is, vaccines, in fact, can work through multiple mechanisms. So what are these mechanisms? So when you receive your COVID-19 vaccine, what will happen in your body is shown there in that yellow graph. And that blip happens within the first few minutes to days of you getting your COVID shot. And what is that blip? And that's what we call the innate immune system. Now, the innate immune system is highly evolutionarily conserved between all creatures on the planet, from Drosophila to slime molds to stockbrokers. Okay, and so the innate immune system is very rapid, highly evolutionarily conserved. This then gives rise to what you call the adaptive immune system. Now, the adaptive immune system is slower acting, slower meaning it takes days, weeks to develop, and it can last for months, perhaps even decades after you get the vaccine. For example, if you get the smallpox vaccine, the adaptive immune response, the system, launches a response that can last for several decades. Now, this adaptive immune system is not highly conserved in all creatures on the planet, but it's mostly in creatures, animals that have developed since the advent of jawed vertebrates. Now, a remarkable feature of the adaptive immune system is what you call memory. Now, you know, our nervous system, our brains can remember an encounter with a person or some event that's happened to us. So too, the immune system has this remarkable feature called memory, in the sense that when you launch the adaptive immune response, if this person encounters the same virus or the same pathogen several decades later, this adaptive immune system can launch an incredibly accelerated and enhanced response, shown there in the blue line. So you see, this is called immunological memory. Now, you might say, how are these various immune mechanisms generated? What are the different cells of the immune system that do this? Now, you can see on the top right-hand side that blue cell out of which there are these orange-looking molecules. These look like a Y-shaped molecule that are coming out. You know, those orange Y-shaped molecules are what we call the antibody molecule. Now, this is the molecule in your body that, if you're vaccinated and if you're infected with the coronavirus, will bind that virus, latch onto that virus, and neutralize that virus and prevent you from getting very sick. Now, below that blue cell, you see another light blue colored cell interacting with the yellow cell. What's that? That is the T cell. The blue colored cell is the T cell. Now, the T cell is interacting with another cell in the body that has been infected by a virus. Now, what the T cell is doing is recognizing that cell that's been infected by the virus and killing that cell. So T cells are really good at searching for infected cells and destroying those infected cells. So you see these two broadly different mechanisms, the antibodies made by B cells, shown there on the top, and the T cells that can search and kill infected cells, represent incredibly complementary and synergistic mechanisms by which vaccines work. So on this slide, you can see I've mentioned to you three different types of mechanisms: the early innate response, the adaptive system, which is memory, and it's comprised of B cells that make antibody and T cells that search and destroy infected cells. Given that is the case, what we have done, and now a number of other groups, is to use what we call systems biology. Systems biology is really an approach to understanding the whole physiological system as a whole, rather than focusing on one aspect of it. And how that's possible is through the advancement of these technologies shown on the right there: genomics, transcriptomics, metabolomics, flow cytometry, and so on, by which we can measure in great detail the behavior of genes, proteins, metabolites, cells, and cytokines in a person. Now, particularly, we can see how these various parameters change in response to vaccination. So if you were to get one of these COVID vaccines, in what way would your immune system respond? In what way would the cells of your immune system change with time? And the genes that they express, how would they change with time? So in this way, can we get a broad kaleidoscopic picture of how the immune landscape is changing in response to vaccination? Several years ago, what we did was an experiment in humans in which we decided to use the tools of systems biology to understand how one of the most effective vaccines ever developed, namely the yellow fever vaccine, was working. Now, the yellow fever vaccine has been licensed several years ago, decades ago. It's been administered to 600 million people worldwide. And in fact, a single immunization with this yellow fever vaccine can give you immunity that can last for 3 decades or longer. So that's a remarkably long efficacy. Now, that certainly is a sort of aspirational goal for any vaccine that we're developing, whether it's against COVID or HIV or malaria or any number of other pathogens. The other interesting feature of this yellow fever vaccine is that a single immunization gives you very strong antibody response and T-cell responses, so the two major arms of the immune system that I mentioned previously. So to us, this yellow fever vaccine represented a gold standard vaccine, so effective, 99% effective, and immunity that lasts for 30 years or longer. Incredible T-cell and antibody response. How does it do it? What's the magic behind this vaccine? Oddly enough, you might think that a vaccine that's been given to 600 million people, we would really know the molecular mechanisms by which this vaccine works. Oddly enough, no, because many of these vaccines were developed through a process of trial and error. And yes, we understand that antibodies are important. Yes, we understand that T cells are important. But the molecular details of exactly which genes are switched on by this virus, which receptor in the immune system recognizes this virus, and so on, that wasn't really very well known. So we thought, why not test this out? Why not do an experiment in humans where we would vaccinate people with this yellow fever vaccine, collect blood samples from these people before you give the vaccine, or day 1, day 3, day 7, day 14, day 21, all the way up to a year, and then analyze the immune response using the tools of systems biology. And what we found was, amazingly, there was a gene signature in a drop of blood. By taking a drop of blood and doing gene expression analysis and working with computational scientists, we could define a gene signature shown there in that heat map through which we could predict who would have a high antibody response and who would have a low antibody response, similarly for CD8 T cells. So what we had defined was a gene signature induced by vaccination within the first few days, 3 days, 7 days, so that's very shortly after vaccination, with which, using machine learning tools, you could predict the magnitude of the T cell response or the antibody response. So this was quite exciting because this hadn't been done with vaccines before. So this really provided the first proof of concept that by taking a gold standard vaccine and applying these incredibly rich technologies of systems biology, you could define biomarkers of vaccine immunity. And it wasn't just that, because the genes themselves were so rich in biological insight. They provided so much information about how this vaccine was working, and we and a number of others have tested this and really gleaned much mechanistic insight about this vaccine. Now, as an extension of this approach, what we have done, and now a number of other groups have done, is to increase the repertoire of technologies that you can use for this style of research. So there you can see, you can probe the immune response at many levels of the biological hierarchy, at the cell level, at the transcriptomics level, at the protein level, at the metabolite level, and even at the level of the epigenome. So in this way, you can construct a multi-scale, multi-response network shown there in that slide. And essentially, so you can use these various technologies to construct a multi-scale, multi-response network and evaluate what are the biological pathways that are driving immunity to this vaccine. The other point I want to mention to you is that we're all interested in durability. Some of you would have already received one or the other COVID vaccine, and we'd all like to know how long are we going to be protected. Are we still going to be protected in 6 months from now, 1 year from now? I mean, it would be ideal if we were protected for decades, wouldn't it? Just like the smallpox vaccine or the yellow fever vaccine that I mentioned. And so, keeping this in mind, what we've done is to use this approach that I mentioned to you, the systems biology approach, to see if you can predict the durability of the antibody response in people. And there on that slide is an experiment that we had done several years ago with the influenza vaccine, the flu shot that we all get. And you can see on the left are folks in whom the antibody response wasn't very durable. By 180 days, the response had waned. But on the right, you can see people whose immune response was quite durable. And what we were able to do by analyzing the changes in the expression of genes 1 day after vaccination, 3 days after vaccination, 7 days after vaccination, and using computational and machine learning tools, is to come up with a biological network, a signature that could accurately predict the durability of this response. And now we're extending this approach to understanding durability of the COVID vaccines that are being approved. Right. Now, let's get back to how these vaccines work. I mentioned to you that they work through multiple mechanisms. You remember the innate response, you remember the adaptive response that seems to last for decades, and you remember this immunological memory, and you remember the B cells and the T cells. B cells make antibody. Now, how exactly do you stimulate the innate response? Why is it that the innate response is stimulated? As it turns out, what happens is there are these things called adjuvants, shown there in red, which are triggers for the innate immune system. So the innate immune system can sense adjuvants. Now, it's a strange word, adjuvants. It comes from the Latin word adjuvare, and that means to help in Latin. And what adjuvants do is to help vaccines to stimulate a potent immune response. They do so by activating the innate immune system. So with this in mind, let me give you an example of an adjuvant. Here's an example in which humans were given the pandemic flu vaccine. This happens to be the bird flu vaccine, which is the H5N1 strain of influenza. One group of humans were just given the bird flu vaccine without any adjuvant, shown there in green. The other group of people received the bird flu vaccine with the ASO3 adjuvant. And I don't need to tell you, you can look at the slides to see the incredible effect that adjuvants have had on the magnitude of the antibody response, but also on the durability. So with this in mind, what we did was to conduct a study in non-human primates, rhesus macaques, to assess different adjuvants for their capacity to augment immune responses to a subunit COVID-19 vaccine that is being developed by our collaborator, Neil King, at the University of Washington. Now, this was a highly collaborative study with those companies shown there. GlaxoSmithKline provided us with this adjuvant called ASO3. Cepic, another company, provided us with SWE. It's a different kind of adjuvant. GlaxoSmithKline also provided with AS37. Dynavax provided us with CpG 1018. All these are different kinds of adjuvants. So what we did was to combine each of these adjuvants one at a time with the COVID vaccine that's shown there being developed, immunize monkeys with this, and ask, what kind of an immune response are we getting? And you can see there the neutralizing antibody response against the SARS spike protein. Look at that. I mean, those are incredibly high titers. Adjuvants really work in terms of giving you a potent response. But not only merely potent responses, but highly durable responses. You see here, after you immunize these monkeys, up to 6 months, the response looks fairly stable. I mean, of course, there is a little bit of a decline, maybe a tenfold decline, but nevertheless, the titers at 6 months are still very high. So this is really encouraging, and based on this, this vaccine is now being tested in humans in phase 1 and phase 2 trials. The other point that we're all interested in these days is, even if you get the COVID vaccine, will you be protected from some of these variants of concern that we've all heard about on the news? You've all heard about this B135 virus lineage from the so-called South African lineage, although I hate to use that term. The virus originated in South Africa. The B117 lineage, so-called UK virus. So we tested this in our experiment. And what did we find? Well, on that graph on the left, you can see how well the antibodies from our monkeys can neutralize the B117 UK variant. And you can see they do very well. In circles on the left are how well they neutralize the original Wuhan strain. On the right is how well they neutralize the one from the B117 variant. On the right panel is the same concept, but now for the B135 South Africa variant. You can see there is impaired capacity for the antibodies generated in these monkeys to neutralize it, but the important point that I'd like you to understand is that the degree to which these vaccines are impaired varies incredibly. So the red lines on the right, there's only a 4.5-fold impairment in their capacity to neutralize the B135 South Africa variant. In contrast, the blue line, the adjuvants that were immunized, this is AS37, in fact show a 16-fold impairment. And in yellow, the CpG alum group showed an 8-fold impairment. So what does this mean? This means that the nature of the adjuvant you use, the nature of the vaccine you use, can have a significant impact on their capacity to launch a response that has broad neutralizing capacity against many viruses. And finally, what I want to tell you about is another way by which the immune system can have breadth of antibody responses. Let's go back to this slide. You remember the innate, the adaptive, the memory. You remember the adjuvant that stimulates the innate response. Now, I told you that memory is, in fact, mediated by these B cells and T cells that have immunological memory. So these are the cells that respond in an accelerated way upon re-encounter with the pathogen. But work over the last 5 years now is providing evidence that the innate system, that evolutionary conserved system that seems to respond very rapidly, and that we all thought was really lasting only a few days, perhaps a few weeks. Now there's emerging evidence that in fact this may also have some form of memory. And really one of the key people who've put forward this concept is my friend Mihai Netia, who's in Holland. Now, what Mihai and others have shown is that cells of the innate immune system, if you stimulate them, they undergo what we call epigenetic changes. And these epigenetic changes can mediate a heightened response a few days later upon subsequent encounter with that same stimulus. Now, you might say, what is epigenetic change or what is epigenetic reprogramming? Well, as you all know, in the nucleus of cells, we have chromosomes, and chromosomes can be tightly wrapped up around histones, shown there in light blue. Those are histone markers. When that happens, the genes are unable to be expressed. However, certain stimuli can result in a loosening up of the histones so that the tightness of the chromosomes being wrapped up is now much looser. So this frees up the DNA that can now be expressed. You see, shown there. So that's in an open chromatin conformation. So what we did was to ask, in response to a vaccine, what are the epigenetic changes that happen in the innate immune system? And we did this using this fancy new technology that was developed at Stanford by my colleagues, P.J. Utz and Purvesh Khatri. It's called EPITOF, that allows you to look cell by cell at millions of cells, at each individual cell in response to a vaccine. What are the changes in the epigenome across the entire chromosome? And what we saw was that when you take a drop of blood from a human being and you look at all the different cell types that are in the drop of blood, you can see that they are so different in terms of how they look epigenetically. More importantly, if you give these people a vaccine, shown there, you can see that there are global changes in the epigenome in these innate cells that I mentioned. And guess what? These global changes can last up to 6 months. That is a very long time based on our prior knowledge of what we thought, how long-lasting we thought that the innate system would go on for. So you might say, well, that's all very well, but what does it mean? Why should I worry about this? As a person who gets a vaccine, is this thing you've described of any relevance to me? And I would tell you, indeed it is, because we asked the question, what happens to these people who have undergone these long-lasting epigenetic changes for 6 months? Now, if you take cells from their blood at 6 months, in this case it was 2 months, and you infect these cells with a completely different virus, not the virus that the vaccine was made against, but in this case it happens to be 2 other viruses, dengue or Zika, which infect blood cells. What did we find? These people controlled the replication of these dengue and Zika viruses much, much more effectively after they had received this vaccine, this flu vaccine, with this adjuvant. That was a remarkable observation because we all thought we're giving people the flu vaccine and we're going to be protected against flu, but now, based on these epigenetic changes, that we seem to be able to control these other, what you call heterologous viruses as well. And you might say, is this control of heterologous viruses really anything to do with these epigenetic stuff that you mentioned? Yes, it is, because you can correlate the amount, how well they control these viruses with the epigenetic changes that we saw. Okay, so this summarizes what I've just said. I think what this shows is a new mechanism, a new way of looking at the innate immune system, in which you can give people this vaccine that we gave, in this case the pandemic flu vaccine, with this adjuvant ASO3, and this then stimulates what we call this epigenetic programming in these innate cells. That then seems to last for at least a few weeks, in some cases even a few months. This seems to confer a heightened degree of antiviral resistance. Antiviral resistance not merely against the original virus, namely the pandemic flu, against which this vaccine was designed, but against these other heterologous viruses such as dengue and Zika. So it's a heightened, broad antiviral resistance mechanism that may last for a few months. So I think what the data that I've shown you would suggest is that the adjuvants can imprint a degree of heightened antiviral resistance in the innate immune system. And this antiviral resistance can last for several weeks and perhaps even for a few months. So previously I had shown you this diagram in which the innate response was, I told you, was very short-lived, typically lasting a few days, perhaps 1 week, if you're lucky, 2 weeks. All of a sudden now we have to revise this in the sense that the innate system can last for several weeks, maybe a few months, and adjuvants can have this by imprinting an epigenetic state on innate immune cells. And this then results in enhanced antiviral resistance against a number of different viruses, including dengue and Zika that I had shown you data for. So let me conclude here. Here we are in the midst of a pandemic. Fortunately, vaccines, many excellent vaccines have been developed and many more are on their way. But I'd like to point out that this is not new. We have dealt with pandemics since the dawn of time, shown there. And so the question is, what are the lessons that we've learned from the COVID-19 experience? I think one lesson clearly is that the speed with which these vaccines have been made has been incredibly inspiring. And I think we should learn from how this can be now done going forward, even during non-pandemic times. What are the lessons there? The other specific example I've given you of this concept called epigenetic adjuvants really, I feel, provides you a complementary way of thinking about vaccines. Typically, when we make vaccines, as we've been doing, we're reacting to a pathogen that's emerged. So in other words, first you identify, it's a SARS-CoV-2 virus, then you get the genetic sequence of that virus, then you make your vaccine. So each and every time you have to be waiting for the new pathogen to emerge. But I would like to suggest that this concept of epigenetic adjuvants and some of the data that I've shown you provides an alternative way where you don't have to react and you don't have to wait for a pathogen to emerge. You can simply have a generic way of stimulating the innate immune system for some period of time, maybe a few weeks, a couple of months, by reprogramming their epigenetic status, as I've shown in the previous slides. And so this concept of epigenetic adjuvants could be quite useful during the time of an emerging pandemic, even as the first cases are being reported and the epidemiologists predict that this has pandemic potential. You could envision immunizing people en masse with these epigenetic adjuvants in such a manner so as to rev up their innate immune system to a heightened degree of resistance to any virus that could emerge. So in this way, you buy time. You are proactive. You are waiting for the conventional vaccines to emerge, which under the best of circumstances could take several months to a year, but several months to a year is way too long. So now you are, in fact, imprinting a heightened degree of resistance as a stopgap measure to give people resistance even as we wait for these conventional vaccines. And so this now raises this concept of this universal vaccine. Why universal? A vaccine that could imprint protection against a number of different viruses. So let me end with this slide, Vaccinology BC and Vaccinology AC, that is vaccinology before Corona. What was vaccinology before Corona? Well, as I said, vaccines typically took years to make, 10 years, sometimes 15 years. Progressively with time, with the advent of new technologies, we've learned how to make vaccines faster and faster. But what's remarkable is the rapidity with which we've made COVID-19 vaccines. So in the 11 months since the virus emerged, actually now it's 13, 14 months, COVID vaccines have been made so rapidly. And that's just been remarkable and inspiring. But I would submit to you that 11 months is 11 months too long because millions of lives lost, hundreds of millions of lives lost, and many people infected and having severe disease. So I would submit to you that having a way in which you could stimulate broad antiviral immunity, even at the beginning of the pandemic, is the way to go. that therefore I believe this concept of epigenetic adjuvants is an alternative way and a complementary way that would allow a degree of immunity to be imprinted in the human population, even as we all wait for these conventional vaccines to be made. Thank you very much.